It is a pleasure to be with you virtually at this very important Congress. I wish I were able to be with you, but of course we cannot. Maybe in the future when COVID-19 is history, we'll all be able to get together again. This presentation is on osteoanabolic therapy for osteoporosis. And I'd like to use our advances in basic science with regard to regulatory molecules and hormone action and show how these advances have led to new therapeutic concepts in osteoporosis. I'm sure that many of you are very familiar with this table of drugs that are available for the treatment of osteoporosis. These are different in terms of their mechanisms sometimes, but in the end, all of these drugs are characterized as anti-resorptives. Namely, they reduce calcium egress from bone. In the yellow shaded areas are the drugs for which there is good evidence for fracture protection, either at vertebral, non-vertebral, and at hip sites. So you can see that many of these drugs fit the criteria of global efficacy. That is, they reduce fractures everywhere. And as I mentioned, by reducing calcium egress from bone, that is by reducing bone remodeling, these drugs prevent the progression of osteoporosis. They stabilize or increase bone density, they maintain trabecular architecture, and they increase mineralization density of the matrix. And these are all very good qualities and lead to a reduction in fracture. And to a certain extent, all of these anti-resorptives meet these goals. But one point that these anti-resorptives do not meet is the ultimate goal, which is to restore microstructure, to take a osteoporotic bone, as shown in the lower panel, and improve it to being less osteoporotic. And maybe at the top of this panel, one of these days, one of these drugs will be able to literally completely restore skeletal microstructure. I'm going to limit my presentation today to the three so-called osteoanabolic agents and give you the evidence for to what extent are we getting closer to that goal. Let us start with an interesting irony. The attempt to improve skeletal microstructure as a treatment for osteoporosis began with the development of a drug that was thought to do the opposite, namely parathyroid hormone. And the reason why this is kind of anomalous is that everyone knows that parathyroid hormone is bad for bones. I showed you this slide in my presentation on primary hyperparathyroidism, and it illustrates the devastating potential of parathyroid hormone to destroy the skeleton. On the left is the famous sea captain, Charles Martel, whose bones were literally disappearing over a very short period of time. And on the right are the radiographic depictions of key skeletal areas of bone that are eroded by parathyroid hormone. And we have shown that even when parathyroid hormone, when primary hyperparathyroidism presents as an asymptomatic disorder, parathyroid hormone is bad for bones. So here we have two depictions of severe osteoporosis on the left and on the right. And the question is, how did we make the conceptual leap from parathyroid hormone as a bad actor when present in excess to parathyroid hormone as a treatment for osteoporosis? And the real clue here was given about 23 years ago, in which it was shown that the way in which parathyroid hormone is administered makes a big difference in terms of whether it is bad for bones or whether it is good for bones. And the daily low dose administration was shown by Harold Dobnik and others, was the way in which parathyroid hormone can be shown to be anabolic. Now, I'm using the terminology parathyroid hormone, but I'm really referring to teriparatide. The teriparatide is the 34 amino acid fragment of the full-length molecule, and it is this form of parathyroid hormone that has been developed as a treatment for osteoporosis. PTH 1 to 84 is not the treatment for osteoporosis. Using teriparatide, Robert Neer showed about 20 years ago in a very well-designed clinical trial that the dose that we're using today, 20 micrograms, substantially reduces the relative risk of new vertebral fractures by 65% and by non-vertebral fractures by 53%. Both of these reductions are highly significant. Dr. David Dempster, a colleague of mine at Columbia, has shown by bone biopsy that teriparatide actually does improve bone quality. In this particular depiction, it improves trabecular connectivity. And I will summarize here the major clinical trials with teriparatide that have shown those reductions in vertebral and non-vertebral fractures. The drug is as effective in women with mild or severe previous fragility fractures. The drug is effective in those who have single or multiple previous fragility fractures. It's effective in glucocorticoid-induced osteoporosis and has been shown to be effective in men with osteoporosis. This drug has been a cautionary note with regard to osteosarcoma that has been shown in rats given high doses for prolonged periods of time. We now have 18 years of experience worldwide on whether or not that safety signal in rats is relevant to us. And very recently, in fact, in the February issue of the JBMR, the 15 years follow-up has been published. After that survey period, there have been no oncogenic signals whatsoever. And it would seem that it is extremely unlikely that osteosarcoma is a human toxicity. And because of that, the FDA in December of last year removed the black box warning for the branded product, Forteo, or I think in Chile it's called Forsteo, but the branded product in the US no longer has the black box warning. Now, a perfect anabolic agent would stimulate bone formation forever, and there would be no increase in bone resorption. This would be kind of a perfect situation. How would an anabolic agent work? There are three potential mechanisms. At the top of this cartoon offered by Dr. Dempster, there is a bone formed on the remodeling surface, and that is called remodeling-based bone formation. And you can see the scalloped line that represents the bone remodeling unit, and bone is formed within it. There is another kind of bone formation in the middle of this slide called modeling-based bone formation, in which bone is accrued on quiescent surfaces. This is the kind of bone formation that we typically see in children, but it is not very common to see modeling-based bone formation in adults. And at the bottom of this slide is the third kind of bone formation, in which there is so much bone formed in the bone remodeling unit that it literally spills onto the quiescent surfaces. And this kind of bone formation is called overflow modeling-based bone formation. Now, with regard to teriparatide, Dr. Dempster showed this at the recent meeting of the ASBMR in September of last year. This was a double-blind, placebo-controlled trial of patients who did not have osteoporosis. They were going to have hip replacement surgery for osteoarthritis. And one group was given 20 micrograms of teriparatide for 6 weeks prior to surgery, and then they were labeled with tetracycline after teriparatide. And at the time of surgery, a sample of the femoral neck was obtained. And in the red boxed areas, you can see that at both the cancellous and the endocortical envelopes of bone, the remodeling bone formation in the middle and overflow remodeling based bone formation on the right are increased over controls, while modeling based bone formation, although greater compared to placebo, did not quite reach statistical significance. And on the basis of this study, it would appear that teriparatide is an anabolic agent by stimulating remodeling-based bone formation as well as overflow remodeling or modeling-based bone formation. So in terms of teriparatide, we see an increase in bone formation markers and then a downturn. We also see an increase in bone resorption. That happens later, and then there is a downturn. And the time difference between the stimulation of bone formation and the stimulation of bone resorption, which is thought to mitigate the anabolic potential of PTH, is considered to be the anabolic window. One would like to open that anabolic window wider to have even a better osteoanabolic agent. Teriparatide stimulates bone formation to a greater extent than it stimulates bone resorption, and this ratio of greater bone formation to resorption certainly helps to define teriparatide as an osteoanabolic agent. Let me now move to abaloparatide. Abaloparatide is an interesting molecule. It's shown in schematic form on the bottom of this slide in comparison to PTHRP in the middle and teriparatide on the top. The green represents the sequences of teriparatide, and you can see that between teriparatide and PTHRP, there is some homology, but there's a lot of white in PTHRP, which means there are many different amino acid residues in that PTHRP 1 to 34 molecule. Abaloparatide looks identical to PTHRP out through residue 22. And you can see in the yellow boxes for abaloparatide, there are different amino acids placed strategically between residues 22 and 34. So abaloparatide is an analog of PTHRP, but different in several important respects. The development of abaloparatide was based on the hypothesis that it would bind more selectively to a configuration of the PTH receptor known as RG. There are two receptor conformations. On the left is the RG, and you can see that teriparatide and abaloparatide have pretty much the same affinity for this form of the PTH1 receptor. On the right, however, there's a difference in the affinity of the other conformation, the so-called R0. And in this conformation, abaloparatide has much less affinity than teriparatide. So if you compare the ratio of affinities of these two parathyroid hormones for these two conformations of the PTH receptor, it is clear that relatively speaking, abaloperitide has greater affinity for the PTH receptor in the RG configuration than in the R0. The RG configuration of the receptor is thought to bind very transiently to the ligand abaloperitide, leading to a burst of cyclic AMP that is only momentary in time. And as a result, on the bottom of this cartoon, there's a greater stimulation of bone formation by stimulating osteoblasts than bone resorption by stimulating osteoclasts. On the other hand, teriparatide seems to have, if you will, more affinity than abaloperitide for the R0, which is a more prolonged experience and leading to a greater, a longer increase in cyclic AMP. And this more prolonged exposure is thought to lead to a more equivalent stimulation of bone formation and bone resorption. Dr. Dempster has looked at abaloperitide in a similar way that I showed you with regard to teriparatide, trying to determine how abaloperitide is an anabolic. And you can see on this slide, just published a few months ago, the top of this slide shows you the various compartments of bone, the cancellous, endocortical, intercortical, and periosteal compartments. And in the orange, you can see that abaloperitide is stimulating the mineralizing surface over bone surface remarkably at all of these four sites. On the bottom, Dr. Dempster has looked at the three kinds of bone formation: remodeling bone formation, modeling bone formation, and overflow modeling-based bone formation. And you can see at the three envelopes of bone, cancellous, endocortical, and periosteal in particular, there's a major increase in overflow modeling-based bone formation, in modeling-based formation, but also in overflow modeling-based bone formation, and also in classical remodeling-based bone formation. Note, however, that the periosteum does not seem to have any remodeling-based bone formation. Looking at this in another way, looking at the remodeling surface on the left at cancellous bone, endocortical in the middle, and periosteal on the right, the purple wedge becomes greater after 3 months of abaloperitide, both at the cancellous and endocortical surface. Looking at the modeling-based bone formation, you begin to see it with cancellous to a greater extent with endocortical, and in the green, you see a substantial amount of modeling-based bone formation in the periosteal surface. And finally, overflow modeling-based bone formation becomes apparent at the cancellous and endocortical sites. So clearly, abaloperitide is behaving like it is an anabolic agent by taking advantage of all three mechanisms of anabolic activity. The clinical trial that led to the approval of abaloperitide is shown here. This was a placebo-controlled blinded study in terms of placebo versus abaloperitide. There was a teriparatide arm, but it was open label. The data show that over the first 18 months of this study, abaloperitide and teriparatide equivalently stimulated bone density and also equivalently reduced new vertebral fracture. In terms of the hip regions, there was, if you will, an earlier and greater increase in bone density of the total hip and femoral neck, and similar reductions in non-vertebral fracture as well as in clinical fracture. Non-vertebral fracture shown here shows that abaloperitide begins to separate from teriparatide early on in the 18-month study. And thus the data are fairly compelling in the first part of this study. This study was also designed to have an extension of 24 months in which the placebo group switched to alendronate, the abaloparatide group switched to alendronate, the teriparatide group ended. So we now have two cohorts, each getting 24 months of alendronate on the backdrop of either placebo or 18 months of abaloparatide. And the analysis of this extension study looked at new vertebral fractures, non-vertebral fractures, clinical fractures, major osteoporotic fractures, as well as bone density. This is the follow-on treatment looking at non-vertebral fractures, and you can see that these are sustained over the entire period of time, including the 24 months of alendronate. Vertebral fractures, non-vertebral fractures, all clinical fractures, and major osteoporotic fractures were all more evidently affected and reduced by the history of abaloparatide use prior to the conversion to alendronate. A very interesting observation in terms of bone turnover markers is shown here. You can see in the blue, which is teriparatide, there is an increase in P1NP as a bone formation marker. That is also true for abaloparatide in the yellow parts of the graph. On the right, while both stimulate bone resorption, teriparatide stimulates bone resorption to a much greater degree than does abaloparatide. And looking at this, one could argue with a similar increase in bone formation markers, but a difference in bone resorption markers with abaloparatide, you in a sense are widening or opening that anabolic window. There were subgroup analyses conducted in this active extend group. There were actually 9 subgroups identified by bone density, by age, by vertebral fracture. And in short, if you look at new vertebral fractures by baseline bone density at any of these 3 sites, the analysis shows that the drug was equivalently effective, independent of what the bone density was at any of these 3 sites. Similarly, the subgroup analysis of bone density at the lumbar spine or total hip showed no difference. Bone density improved to the same extent, independent of what the starting T-score was. And in fact, in the majority of the subgroup analyses, abaloparatide performed better than placebo cohorts. I was part of a study that looked at one of these groups, namely renal function, dividing the active trial into those with reduced clearances of 30 to 60, and then reasonably normal of 60 to 90 or greater than 90. And in terms of changes in bone density, this slide shows that the lumbar spine, hip, and femoral neck, there was essentially no difference among these 3 different groups of different kidney function. So the drug seems to be very effective, independent of renal clearance. In terms of side effects, there was a greater incidence of hypercalcemia as a pre-specified endpoint in the teriparatide group than in the abaloparatide group. In terms of cardiovascular elements such as dizziness and palpitations, there was a somewhat greater incidence among the abaloparatide group than the teriparatide group. Very recently, Felicia Cosman has published in the JCM an analysis of heart rate. The top part of this figure is maximum post-dose heart rate versus placebo. And you can see there's really essentially no difference between abaloparatide and teriparatide, with a slightly increased heart rate with both of these drugs. On the bottom of this slide is a comparison of the maximum change from pre-dose to post-dose heart rate, and you really don't see any major difference between abaloparatide and teriparatide. Comparing teriparatide and abaloparatide, Abaloparatide does not require refrigeration after being first used. They're both daily injectables, and the dose is different. In terms of duration of therapy, in the United States, the 2-year limit has been lifted for the branded product of teriparatide. It continues to be limiting for abaloparatide. Maybe the anabolic window is wider with abaloparatide. I think the adverse event profile is similar between the two, and I've already covered the theoretical safety concerns in terms of osteosarcoma. So filling in this table a little bit more, abaloparatide, I think, stimulates bone formation to an equivalent degree, but not stimulating bone resorption to the same, giving a ratio of bone formation to resorption that is greater than teriparatide. Let me now move to romosozumab. Romosozumab was developed after observing two very rare diseases, sclerosteosis and Van Boeken's disease, in which patients accrue bone density for their entire lives, but they never fracture. It is due to the absence of sclerostin, which is an inhibitor of one of the major anabolic pathways of bone, the so-called Wnt pathway. The problem with these two rare diseases is that the apertures of cranial nerves get compromised and they develop severe neurological complications over time. Among heterozygote carriers, however, they also have high bone density, but they don't have those neurological features. Again, the sclerostin gene is compromised, but only halfway. And it would seem that this might then be a clue to a therapeutic drug. Let me explain this a little bit more. This is the pathway where Wnt engages with LRP4, 5, and 6, and by doing so prevents the intracellular metabolism of beta-catenin, allowing it to enter the nucleus and transcribe anabolic features of bone. With sclerostin, it interposes itself between Wnt and these transcription factors so that beta-catenin is metabolized in the cytoplasm, and thus the anabolic potential of Wnt is compromised. In terms of an antibody against sclerostin, what happens is that sclerostin gets bound by the antibody, then allowing Wnt easier access to its ligands and facilitating the anabolic potential of this pathway. The clinical trial of the drug, known as romosozumab, an antibody against sclerostin, was published by my colleague Felicia Cosman, in which romosozumab was compared against placebo, monthly dosing for 12 months, followed by denosumab for the next 12 months. The endpoints were fractures at 12 months, that's romosozumab alone, and at 24 months, which was romosozumab followed by denosumab. And on this slide, you see the major changes in bone density. At 12 months, I've shown with the red arrows, major improvements at the lumbar spine, 13% after 12 months. And after denosumab, 17% increase after 24 months. There was a 73% reduction in relative risk of new vertebral fractures, a 36% relative risk reduction in clinical fractures after 12 months, and after 24 months, there was a 75% relative risk reduction in new vertebral fractures. Kenneth Sag has followed on a study comparing head-on romosozumab with alendronate, followed after 12 months with both groups getting alendronate. And again, you can see at the top, bone density goes up remarkably with romosozumab compared to alendronate. And at the bottom, you can see this very interesting pattern of bone turnover markers with an initial burst of bone bone formation with P1NP, but never an increase in CTX. In fact, it goes down and follows even further down below baseline after 12 months. Comparing romosozumab with alendronate at 24 months, the group that initially received romosozumab continues to do better with regard to relative risk reduction of 48% compared to alendronate after the 2 years. And it's not just new vertebral fractures, it's clinical fractures, it's non-vertebral fractures as well, with romosozumab showing greater efficacy when it is preceding the use of alendronate. The effect of romosozumab supports a modeling mechanism. You can see bone formation rate at the periosteum, the endocortical compartment, as well as these other compartments are markedly stimulated in this model of the overrectomized rat treated with the sclerostin antibody. And work from David Dempster and Michael Ominski have shown that using the antibody at either the lumbar spine vertebrae or proximal tibia, you see the labels are uniformly seen in comparison to vehicle, thus giving good evidence further that one of the mechanisms by which this anti-sclerostin antibody works is by a modeling base. Also noteworthy, and I pointed this out earlier, there does seem to be an anti-resorptive effect with this anti-sclerostin antibody. And again, shown here is this very curious anti-resorptive effect of this anabolic agent. So going back to our cartoon, we might actually look at this and say, well, bone resorption is reduced, bone formation is increased, and this anabolic window is actually much wider with romosozumab than with the other two osteoanabolics. So filling in this table, we have bone formation increased with romosozumab, bone resorption decreased, so that the ratio of formation to resorption is even greater with romosozumab in comparison to the other two anabolics. Now, I didn't mention about safety, but let me make a point about safety. There was a imbalance in cardiovascular events with romosozumab in the trial in which alendronate was compared with romosozumab. There was no imbalance in cardiovascular endpoints in the pivotal clinical trial when romosozumab was compared to placebo. It is not clear whether this imbalance in some cardiovascular endpoints was due to a true increase in the serious side effects with romosozumab, or whether it was due to a reduction in expected events in those who received alendronate. And because that study did not receive a placebo control, it was impossible to be sure. And because of this uncertainty in the United States, romosozumab has been approved with a black box warning. And we really need more information in order to really understand better whether or not this imbalance is due to the drug itself or due to a relative good, if you will, with regard to alendronate. So let me summarize what I have covered with regard to osteoanabolic therapy. New insights into bone biology have led to new osteoanabolic approaches to the treatment of osteoporosis. The three osteoanabolic agents I've covered, namely teriparatide, romosozumab, and the valoperitide. Together, these three osteoanabolic agents have broadened our views of mechanisms by which the skeleton can be strengthened in osteoporosis. I think it is a nice lesson to be learned that advances in our understanding of basic bone biology led so quickly to the advent of an approach to treatment that goes beyond the anti-resorptive motif, but now includes the possibility that we can not only build bone by improving bone density, but we can actually build bone quality by helping to restore skeletal microstructure. So I, again, enjoyed very much being with you today, and I am very pleased and honored to be a participant in this very important Congress. Thank you very much.